Hello, can someone please confirm you can hear me?
We can hear you.
Thank you. All right, before we begin, are there any questions? You should be seeing my screen now. It's not coming up.
Wait, I had a question.
Uh-huh.
For the quiz, does the chapter one study guide include the section of chapter 10 that was covered in the slideshow?
No, there's a study guide for chapter 10. It is in the section called, what is it? To the objectives and overview of chapters 1 and 10, but it's a separate
Oh,
section.
okay. Also, is the quiz multiple choice, just like the practice quiz?
Yes.
Okay. And also, does the quiz include, um, the only includes the first half of the chapter two, correct?
Let me check on that.
Okay.
Just give me a minute here. I'm trying to find my place in the slide
Okay.
because it didn't open to that. Well, I've got here. I don't know if that's correct. Is that correct? We were on slide 29. Let me see if there's another one. Yeah, that's the only one. All right. Um, let's go to the syllabus. And the question was, what part of chapter 2 is on quiz 1? It's about where we left off on chapter 2 last time. So I
Okay.
don't know if that's half the chapter or not, but we are on what slide are we on? 29. So you should know at least carbohydrates, if not the start of lipids and everything before that on chapter 2.
Okay. Thank you so much.
All right. All right, let me go to the syllabus again. So today is October 7th, and we're supposed to be working on chapter 2 and finishing chapter 2. Hopefully, we'll finish chapter 2. I don't know if we'll succeed. Let me warn you, you do have a quiz, quiz 1, this Wednesday. We're covering chapter 1 and chapter 10. And chapter 10 is only what we covered in chapter 1, the start of chapter 2, and the labs 0, 1, and 2. Any questions about anything so far? There is a lab today. We'll talk about that in the lab time if you haven't had the lab yet. So let's move on to chapter 2. I think I'll leave this up just in case.
For the quiz, can you use pen and pencil and paper or no?
It's online. You could print out the question and then answer it with pen and paper, and then put your answer digitally in the quiz online, but it's an online quiz.
Oh, okay.
Does the quiz have two chances like the practice quiz did?
No. Each quiz you only have one chance on. So you should know the material before you take the quiz. I thought I stated in the practice quiz and the environmental quiz that these quizzes were unusual in that I was giving you two chances. All right, so we discussed about phospholipids. Can somebody confirm that?
Yeah, we did.
All right, so let's finish up the lipids. Besides phospholipids and fats or triglycerides, which are made up of monomers, the fatty acid chain and glycerol, and then the phospholipid also has a phosphate group on it. Those are the monomers making up these lipids. There are also lipids that are not polymers. There's one molecule and they're not a polymer made up of monomers. And these are the steroids and the sterols. A steroid is 4 interconnected carbon rings, like this. Characteristic of this group, if there's an OH group attached to the sterol, we call this, it's steroid, we call this a sterol. Cholesterol is the most famous sterol. Sterols are an important part of the animal plasma membrane, where, let me blow the slide up, we have in our animal cell membrane cholesterol inside the membrane. And it's just in there between the phospholipids. And that cholesterol gives strength and support to this membrane. The reason why animals have it is because they only have a cell membrane. They don't have a cell wall to help protect the cell membrane. So animals put cholesterol in their cell membrane. And if you don't know, let me inform you, you can only find cholesterol in animal tissue. Plants, as well as, microorganisms like bacteria, do not make cholesterol. Only animals do. And so if you want to have a cholesterol-free diet, you have to eat a plant-based diet. Unless you have like processed milk that doesn't have cholesterol in it, which I guess skim milk probably would have very low amounts of cholesterol in it. All right, any question about lipids? Let's move on to the third biological molecule you can find in life of the 4 biological molecules, and that is proteins. Proteins are an essential in cell structure and function. Proteins may be structural proteins found in bone, muscle, and hair. They may be enzymes which speed up chemical reactions. They may be transport proteins which move chemicals across the cell membrane. They may be regulatory proteins which regulate body functions like a hormone, like children make the growth hormone that helps them grow. Proteins may also be responsible for movement, the reason why we walk, the reason why my arms are moving, even the reason why a bacteria can swim is because of proteins. We have proteins which contract and it allow the muscle to move, and the protein contracts to make the bacteria, what do you call that? The flagella move. There are also proteins responsible for some bacterial toxins. Some bacteria make a toxin, which is something that's toxic to us. And some of these, actually many of them, are proteins. And then we have proteins which we call antibodies. And these are proteins that we make or an animal makes to help defend the body against invasion by a microbe. Any question about any of these different functions and structure of the proteins? As you can tell, proteins are a very diverse group, and they differ more among each other than probably any other of the biological molecules. And there's a reason for that, and that is proteins are a polymer, and they're made up of monomers, but there are 20 different monomers, at least in humans, and in many living organisms, there's 20 different monomers that make up the polymer. And when we're talking about carbohydrates, we might have a handful, a dozen at most, carbohydrates that make up, I should say that, monomers that make up the polymer of a carbohydrate. For lipids, we have a handful as well, maybe as high as a dozen. And let's see, for nucleic acids, we only have 4 monomers, which make up the nucleic acid. And proteins are more diverse and they have more building blocks, 20 different building blocks to make the protein. Proteins consist of monomers that we call amino acids. Each amino acid has an amino group on it on one side, and then a carboxyl group on the other side. And this is a generalized structure of amino acid, having a central carbon, an amino group to one side, a carboxyl group to the other side, a hydrogen, I guess to the third side, if you want to call it a third side. And then on the fourth side, we have a side group that we call R, and R just stands for something, and R can vary, and there are 20 different R's in humans because we have 20 different amino acids, and the R group varies with each amino acid. So tyrosine, for example, the R can be this structure here, and obviously the R group depends on which amino acid you have. Many amino acids are linked together to form a protein. Here we're looking at some of the amino acids. You don't need to know this slide, but the simplest amino acid for the R group just has a hydrogen, and that's glycine. Alanine has a methyl group attached to it. Valine, 3 methyl groups attached to it. Leucine, 4 methyl groups attached to it in this type of a structure or a branched chain. And then isoleucine has this type of a branched chain. Phenylalanine has this structure attached to it. That's not phenol, because that's phenol here, but what the heck is that called? Phenol carboxylic acid, but that's the old term. What the heck is that name?
Is it just an aromatic or benzene?
Yeah, benzene. Thank you. I was trying to remember what it's called. Benzene. Carboxylic acid, same thing. This is phenol, unless phenol is carboxylic acid. I don't remember. Anyways, one of these is carboxylic acid. This part here is phenol, meaning it has an OH group to it. And this one, tyrosine, has a CH2 connecting the central carbon to the phenolic group. And then there's this type of structure, that type of structure, that type of a structure. Some of these groups are, some of these R groups are nonpolar, like this is a nonpolar group, and all of the previous ones, all of these are nonpolar R groups. But some of the R groups are polar, like this one, the OH group is polar. Obviously, this part of it is not polar, so that part is nonpolar, but that is polar. And then this is polar here and there, so this one is polar. That one has a little bit of polarity to it. This area here is polar, and that one is nonpolar. You don't need to memorize these different amino acids. I'm just showing you. And let me state that all amino acids are both polar and nonpolar, and at least in a region. Some of them are almost entirely polar, like that one here is pretty much polar. But they're all polar, even the ones that have a nonpolar R group, because the amino group and the carboxyl group are both polar, and that's a nonpolar region. So proteins are both polar and nonpolar, at least in the R group. Some of the R groups are nonpolar. And that one we call polar, and that one is polar. So these amino acids are considered to be polar amino acids. Any question about any of that? So even though this one is nonpolar for here, and then this structure here, because of that, we call this a polar R group. Any question about any of that? All right, so the big point is that proteins are both polar and nonpolar. When the protein is around water, the nonpolar groups will be tucked in the protein, and then the polar groups will be on the outside of the protein facing the water. If, on the other hand, the protein is in a lipid, like a protein in the cell membrane, it'll have the nonpolar regions on the outside. At least the nonpolar regions will be facing the nonpolar lipid membrane, but the polar regions will be tucked on the inside, so they're not exposed to the nonpolar part of the lipid membrane. And that's just the way proteins are. Any question about any of that? You get many amino acids linked together to form a protein. And how they link is by a peptide bond. A peptide bond is when, let me blow this up. Water comes out, meaning OH from this amino acid, H from this amino acid, and then that nitrogen bonds directly to this carbon here in dehydration synthesis. And this bond between nitrogen and carbon is rare, not only in biological molecules, but it's rare in the universe. And that's why it's called a peptide bond, because we do find it in peptides or polypeptides like a protein. Anyways, you link these amino acids together with the peptide bond, one after the other, and then you get a polypeptide. A polypeptide is just a chain of numerous amino acids hooked together. If it's a short chain, it's called a peptide, but if it's many amino acids, you call it a polypeptide. And a growing protein is a polypeptide until the last amino acid is added. And only after the last amino acid is added do you call it the finished protein or the protein. Any question about any of that? So while the protein is growing, you can call it a growing protein, but its correct name is a polypeptide. And then once it's added the last amino acid, you call it the protein. Any question about any of that? All right, in a dehydration synthesis, you pull out the OH and the H group, making the peptide bond between the amino acids, and then water comes off. There are different levels of protein structure to a protein, and there are 4 possible levels of protein structure: the primary level, the secondary level, the tertiary level, and the quaternary level. Let's watch a little video on the different levels, the 4 levels of protein structure. It looks like we have a
ChatGPT is great for quick answers, but when you need writing expertise, Grammarly is my go-to tool. If you're already...
In this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consisting of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein.
All right, let's talk about the 4 levels of protein structure. Someplace around here.
Which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's two of them that you need to be familiar with, the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by
Just
hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid.
trying to stop the video here and I got the wrong button. All right, let's come back to the secondary structure. It said it was stabilized by the hydrogen bonds. It is true that there are hydrogen bonds in the secondary structure, but there are a lot of other bonds in the secondary structure. And I'll talk about them in a little bit, but just let me warn you, this video is oversimplified the secondary structure, stating that the secondary structure is stabilized by hydrogen bonds. It's also stabilized by ionic bonds, ionic repulsion, van der Waals forces. I'll talk about those in a little bit. And ionic attraction, ionic repulsion, and then repulsion between a polar molecule and a polar amino acid and a non-polar amino acid.
You can
Professor, we can't see your screen.
see
Oh, you can't see the
by
screen. Sorry. Thank you.
hydrogen bonds.
So it's more
The NH group of one amino acid interacts with the residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid.
okay. It can be stabilized by other attractions, mainly ionic attractions, but the hydrogen bonds are probably the main one in the beta pleated sheet.
Between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions about that video? Come on. Sorry, I'm having trouble getting that to work. There we go. So there are 4 levels of protein structure. Let's talk about the primary level first. The primary structure is just the specific sequence of amino acids in a polypeptide chain, one after the other. When the protein is first made, all it has is the primary structure. One amino acid followed by next. However, once the primary structure in the protein is made, nearby amino acids start interacting with each other. There can be attractions, like right here. We have a hydrogen bond between this amino acid and that amino acid. There can be van der Waals forces, meaning this amino acid is nonpolar and it's attracted to this amino acid here. A van der Waals force may be something you learned in the preliminary biology course. Van der Waals forces are attraction between nonpolar molecules. Like when you have oil mixed together in a bottle, the reason why the oil is a liquid is because the oil molecules are attracted to each other. It's a very weak attraction called van der Waals forces. And the reason why it's a liquid is because the van der Waals forces hold those two molecules together before they split, and then they come back together or come back together with another one, and that's why it's a liquid. You can put like olive oil at room temperature, and the van der Waals forces keep the olive oil a liquid. But if you put that olive oil in the refrigerator, it will cause the van der Waals forces to be locked in place, and then the two molecules will not break apart, and that will have solid olive oil. Any question about that? And then you can heat up the olive oil, and you'll totally break the olive oil attraction from each other, and then you won't have liquid olive oil, you'll have gaseous olive oil. So van der Waals forces with oil are an attractive force holding the molecules together to make it a liquid, which is why olive oil, at least at room temperature, is a liquid. Any question about that? Anyways, they're very weak, but they can be forces of attraction between two nonpolar molecules. And then you can have forces of repulsion. If that is a nonpolar amino acid, and that's a polar amino acid, they will be repulsed from each other. And then this one here, let's say this is a positive ion, meaning this amino acid has a positive 1 charge, and there's the same amino acid with a positive 1. That would be a force of repulsion between these two amino acids. And then you can have ionic forces of attraction, which I don't know. Let's go ahead and talk about these maybe. This is a positively charged amino acid, meaning it has a +1 charge, and that is a negative amino acid, meaning it has a -1 charge. That will be a positive force of attraction, and that will be even stronger than the hydrogen bond, because that's an ionic attraction. Any question about any of that? The key to remembering is that the secondary structure is localized interactions between neighboring amino acids. So all of the amino acids being attracted to each other will be neighboring amino acids, one nearby the amino acid. And that gives rise to the secondary structure. And these forces are hydrogen bonds, hydrophobic interactions, could be hydrophobic attraction, could be hydrophobic repulsion, meaning hydrophobic versus hydrophilic. And it could be ionic bonds, either an ionic bond forming between them or a ionic repulsion if they both have the same charge. Any questions about any of that? All right, the video showed you the beta pleated sheet showing you here. And then excuse me, there's the beta pleated sheet. This is the alpha helix. And this is the beta pleated sheet. And this is the linking of the amino acids. And then they're held together right here by hydrogen bonds. This one to that one. There could be an ionic force of attraction as well in the beta pleated sheet. Usually forces of repulsion are not playing a role in the, you don't need to know that in the beta pleated sheet because they're being held together in the beta pleated sheet. The third one, which isn't shown here, and he did actually show it, is the random coil. And the random coil can have places which are attracted together and places which are repulsed from each other, and we'll talk about that in the tertiary structure. The tertiary structure is the overall three-dimensional structure of a single polypeptide chain. This occurs when the helix of a protein folds irregularly, forming the I don't know, the knotting of the protein, the three-dimensional structure of the protein. And this can happen by all of the forces of attraction and repulsion that we saw in the secondary structure, meaning hydrogen bonds, hydrophobic interactions like hydrophobic forces of repulsion, ionic forces of repulsion, ionic forces of attraction. What am I missing? Oh, the van der Waals forces. So you see all of those in the tertiary structure, as well as one other force of attraction, and that's the disulfide bond forming between two cysteine amino acids. So we have a cysteine here and a cysteine here. And let me go ahead and show that out here. Paint. So we have a protein here, and we're going to make it over here. And these are forces of attraction right here. So we're just going to put a line there, which I probably should do a dotted line instead to make it clear that's a force of attraction, except it's very hard to make a dot. As you see, I just made a squiggly line. That's awfully small. Where do I get the icon? Is that it? There's the brush. All right. All right, the important part is there's forces of repulsion here, so these are moving away from each other. And then there can be a cysteine here. I had a hard time writing that, but that's all right. Cysteine has a SH group. Oh, let me just write that. It would be too hard to. And there's a bond between the cysteine and the sulfur and the hydrogen. And then we're going to make another over here. Oop. And let's go ahead and draw that there. And make a bond here. Oh, I don't have the right thing. Between this cysteine over here. Having a hard time doing the Y. There we go. So that's the cysteine right there. This is the protein right here. And then we're going to come together again on the protein. So there's forces of attraction, and then this holds together, which I kind of got off the screen, but you see, oh, there we go. Didn't even see that. So this would be a region which is attracted to each other, and so we're going to put some hydrogen bonds, ionic attractions, whatever. In this region, what's going on here? I can't draw that. There we go. And then there would be forces of repulsion here, and that's why these are away from each other, these amino acids here. All right, the question about the cysteine is those hydrogen can come off, and I don't know how to get rid of that, so let's just, oh, maybe I can erase that. I don't know. Yes, I can. The hydrogens come off and go into solution, and then the sulfur groups form a covalent chemical bond between them. This is a very strong bond, much stronger than the hydrogen bonds, and that's what's holding the amino acids or the atoms and the bond of the protein here. Oops, don't want that there. An example of a disulfide bridge forming is when you have your hair and then you get your hair permed, what they do is they add this link between two cysteines so that the hydrogen comes off and then the sulfur dibridge forms, creating this disulfide bridge, which makes your hair a perm. And this perm can last a month going in the shower. And that's very different from a hydrogen bond. My mother used to get her hair and put it in curls, put a bobby nip to hold it in place, do that for all of her hair, and then put the hair curling cover on, whatever you call a cap on, and then turn on the machine, which would put moisture and heat into the hair. And then she would stop it after, I don't know, 10, 15, 20 minutes, and then take off the cap, take out the curls, and she'd have this nice curl. How that curl formed was it formed hydrogen bonds between the hair, and she'd have this nice curl until she went out into the Western Oregon weather, which had a lot of humidity in it, especially from the mist and the rain, and her curl would then sag. First time she got her hair in very moist air. And that's because that moisture got in between the hydrogen bond and then broke the hydrogen bond. Much weaker than the sulfur dibridge, which makes the perm. Any question about any of that? Anyways, that's the link between the tertiary where are we on this one? Structure that you do not see in the secondary structure, and that's the disulfide bonds, linking two cysteines amino acids together, a very strong force of attraction. The important part to know is the tertiary structure is any amino acid can bind to any other amino acid or interact with any other amino acid. For example, this amino acid at the end of the protein is interacting with an amino acid being attracted to an amino acid near the beginning of the protein. In the secondary structure, we only had nearby amino acids interacting with each other. And then this one would be a force of repulsion between this amino acid and that amino acid. So that's the difference between the tertiary structure and the secondary structure. And there's two differences. and that is the secondary structure, the interaction between amino acids, whether it's attraction or repulsion, happens only with nearby amino acids. In the tertiary structure, it can happen between any amino acids in the protein chain. And then the other difference is the tertiary structure has a disulfide bond being a force of attraction. The secondary structure does not have that disulfide bond. It has all the other forces of attraction and repulsion, but not the disulfide bond. All right, for many proteins, the highest level of structure they get is a tertiary structure. Like every enzyme I know is only made up of one polypeptide chain, like catalase. Trying to think of another enzyme, catecholase, lactase. All of these enzymes only have one polypeptide to make up the final protein. And so the highest level of structure they can get is a tertiary structure. However, if the protein is made up of more than one polypeptide, then we can get a quaternary structure. The quaternary structure is when the different protein subunits, meaning the different polypeptide chains, come together and then form the final protein. And there can be forces of attraction and repulsion between each of the polypeptide chains, which make up the quaternary structure of the protein. Like this one right here, all of these amino acids are being attracted to these amino acids right here. And it looks like this amino acid right there is repulsed from this amino acid right there. Any question about any of that? So you get the same forces of attraction and repulsion in the quaternary structure that you saw in the tertiary structure. The only difference is the quaternary structure is when the protein is made up of more than one polypeptide unit to make up the final protein. And some examples of that are hemoglobin, insulin, and antibodies. Hemoglobin and antibodies are each made up of 4 polypeptides that come together to make the final protein. And so these proteins have a quaternary structure. Any question about any of that? All right. Proteins have an overall shape. There are globular proteins, and these are basically compact, roughly spherical proteins. Hemoglobin is an example of a globular protein. And there are fibrous proteins. These are thread-like, like the protein in collagen and hair are thread-like fibrous proteins. Proteins can be conjugated with other molecules, and we call these conjugated proteins. A combination of the amino acid protein part with other organic or inorganic components. These conjugated proteins are named by their non-protein component. Like a glycoprotein is a protein hooked to a glycogen, and glycogen is a carbohydrate. And you find glycoproteins in the blood. There's also lipoproteins, and this is a protein combined with a lipid. You can find these in the blood as well. When you have a functional protein that's acting properly, it's going to be in a 3-dimensional shape. And you can lose that 3-dimensional shape by breaking hydrogen bonds where they should be there, or breaking ionic bonds where they should be there, or forming hydrogen bonds where they should not be, that can change the 3-dimensional shape of the protein. And when the protein no longer functions, totally non-functional, we say it is totally denatured. And if it's functioning at less than optimal rate, we say it is partly denatured. A protein has to be in the correct temperature, the correct pH, and the correct salt concentration before the protein can have the correct shape, forming the correct attractions and repulsions, and having the correct three-dimensional shape. If you change the temperature, the pH, or the salt concentration, you can get the protein to lose its three-dimensional shape. And initially, it'll become partially denatured. And then later, when it's really at the incorrect temperature, it can become totally denatured. Any question about any of that? Whoops, wrong way. All right, any question about proteins? So to have the protein at its optimal functioning rate, it must be in the correct temperature, the correct pH, and the correct salinity. And proteins are more complex than the other biological molecules, and it's mainly because they have more building blocks, more amino acids than any other carbohydrate or any other biological molecule. They have more monomers. All right, let's talk about the fourth of the biological molecule. These are the nucleic acids. There are two, DNA and RNA, and you can call DNA deoxyribonucleic acid and RNA ribonucleic acid. All nucleic acids consist of monomers that we call subunits or nucleotides. A nucleotide itself consists of three parts. There's a 5-carbon pentose sugar, shown right here. It is deoxyribose for DNA and ribose for RNA, at 5-pentose sugar. And there's a phosphate group attached to the sugar, and then a nitrogen-containing base also attached to the sugar. The nitrogen-containing base can be a purine or a pyrimidine. This is a purine right here, and that's adenine, and thymine is a pyrimidine. There are two purines in DNA, that's adenine or A and guanine or G. And there are two pyrimidines in DNA, cytosine or C, thymine or T. And I don't care how you know the names, you should know DNA has 4 nucleotides, either A, G, C, or T, or you can call them adenine, guanine, cytosine, or thymine. I don't care which. How you can know a pyrimidine from a purine is if the name has a Y in it. Pyrimidine has a Y in it, and everything that has a Y in it in DNA is a pyrimidine. If it lacks the Y, then it is a purine. Now for RNA, we do not have thymine, we have uracil. And I just remember uracil replaces thymine in RNA. Thymine has a Y in it, so uracil is also a pyrimidine because it replaces thymine in RNA. Any question about any of that? In DNA, the bases bind to each other by hydrogen bonding, and the A and the T are complementary, or T and the A, doesn't matter how you word that, for in DNA. So you always find an A versus a T, and there are 2 hydrogen bonds between each A and T. You can also have a C and a G as a complementary base, or G and C, doesn't matter which. And it will hydrogen bond, so a cytosine will always have a G on the complementary base of DNA. And there are 3 hydrogen bonds between cytosine and guanine. Let me see if I can show you that. kind of here. Let me blow this up. So it's showing you 2 hydrogen bonds between A and the T and 3 hydrogen bonds between the C and the G. Any question about any of that? So A and T are always complementary, and C and G. Now remember, for RNA, we have U instead of T. And when the RNA is first being made, it will bind to the DNA, in which case A and U bind to each other. But remember, in RNA, I think I've talked about this, that the RNA binds to the DNA only at the beginning, when that base of the RNA is first put in. After the polymerase moves down a bit, I don't know how many, but 5 to 10 bases, that RNA falls off the DNA, meaning the A and the U are no longer complementary because the RNA has fallen off the DNA, in which case the DNA starts snapping back together. The A and the T will recombine from the other one strand. They're separated, and when the RNA falls off, the other strand of DNA can then come back and complement the first strand of DNA. Any question about any of that? All right. DNA has the sugar deoxyribose, and most of the time DNA is double-stranded, as shown here. You can get DNA single-stranded, though, whenever the DNA is being replicated or whenever the DNA is being transcribed. That portion of the DNA will be single-stranded. There's a sugar phosphate backbone. Let me blow this slide up. And that means that the single strand of DNA is held together by the phosphate group binding to the sugar, binding to the phosphate group, binding to the sugar, and the sugar phosphate backbone is holding the single strand of DNA together by covalent chemical bonds. The bases do not hold the DNA together, at least a single strand of DNA. The base is off on the side, and then the bases come together to make the steps of the helical staircase, and the bases hydrogen bond with each other, holding the 2 strands of DNA together. So 2 strands of DNA are only held together by the hydrogen bonds between the complementary bases in each strand. Remember, hydrogen bonds are weak and they can break and reform all the time. So every once in a while at 37 degrees, this hydrogen bond right here will break, but the DNA is held together by these 2 hydrogen bonds in the guanine and cytosine, these 2 in the A and the T, these 2 more in the A and the T, these 3 in the G and the C. So all the other hydrogen bonds can be holding the DNA together when that one breaks, and then this one will reform, and maybe that one will break. DNA at 37 degrees, the hydrogen bonds break and reform all the time. And we actually call this when the DNA breathes, when the hydrogen bond breaks and then reforms. Like I said, DNA, the 2 strands wrap around each other like a twisted double helical staircase, with the bases being the steps of the staircase. Any question about any of that? Okay, RNA has the sugar ribose. RNA is single-stranded, at least most of the time in the cell, and RNA by itself is single-stranded. The exception is when the RNA is first being transcribed. Something like about 10 bases of RNA will actually hydrogen bond to 10 bases of DNA. But then when the next RNA is added, the oldest RNA, which is bound to the DNA, will then fall off the DNA, meaning that only a portion of that RNA will be hydrogen bonding to the DNA. So RNA is single-stranded. Once again, the sugar phosphate backbone holds the RNA molecule together. The bases of RNA are A, C, and G, the same three as in DNA, but it does not have thymine, T. It has U or uracil. But once again, there are 4 nucleotides that make up our RNA. There are 3 major kinds of RNA. There's tRNA, also called transfer RNA. There's mRNA, also called messenger RNA. And there's rRNA, also called ribosomal RNA. Each of these RNAs plays a role in protein translation. And we'll talk about these when we talk about transcription and protein translation. But let's, for now, just say there are 3 major kinds of RNA. Any question about any of that? All right. ATP is also called adenosine triphosphate. It is the primary energy source for cellular activities. So when a cell needs energy, the cell will most likely get that energy from the molecule of ATP. It is true the cell can get the energy from other molecules, but most of the time it gets the energy it needs from ATP. So we say that ATP is the energy currency of cells. Currency is what you use to get things you buy, and when the cell needs energy, it gets its energy most of the time from ATP. ATP has ribose in it, right here. There's adenine in it, right here. And then 3 phosphate groups right there. The third phosphate group, having a squiggly line holding the P to the O, is a squiggly line because that's a high energy bond. The second phosphate group also has a squiggly line because it's a high energy bond. If the cell needs energy, it will break this high energy bond, releasing a lot of energy, using some of that energy for its energy needs. Some of the energy will be lost as heat. We'll talk about that later. But if it needs energy, it will break off that third phosphate group. And if it needs further energy, it can actually break off the second phosphate group and use the energy from that second phosphate. That energy is just kept in the electrons holding the covalent bond together, meaning this bond right there, and the cell can break that bond, releasing the energy for the cell's energy needs. Any question about any of that? All right. ATP is made by dehydration synthesis. What you can do is get ADP, that's adenosine with 2 phosphate groups attached to it, add on an inorganic phosphate. That's just a phosphate right there. It's more than phosphorus. It's phosphorus with attached to 3 oxygen molecules. And then you need energy to come into the system for this reaction to go. And then we go this way, forming water. And then we combine that phosphate group, making ATP. So ADP plus phosphate and energy can make ATP, and then water. That's how the cell makes ATP, and then the cell can break down ATP to release the energy for its energy needs just by adding water. going this way to become ATP, releasing that third phosphate, and then releasing a lot of energy, which some of that energy can be used by the cell for its energy needs. All right, any questions about any of that? No questions? All right, here's a question for you. In this lesson, what molecules have we discussed that have hydrogen bonds that give the molecule important properties? So name a molecule that we've discussed in this lesson that has hydrogen bonds giving the molecule important properties.
Proteins.
Proteins have hydrogen bonds attracting the amino acids together, which help make the protein form its correct shape. What's another molecule that has hydrogen bonds that give the molecule important properties?
DNA or RNA.
Yes, DNA in particular, you need the hydrogen bonds to hold the two strands together. RNA has hydrogen bonds, but mainly when the RNA is first being transcribed and the hydrogen bonds of the RNA will attach to the DNA to make the RNA and the DNA double-stranded, at least for a little bit. But after a while, that hydrogen bond will break and then the RNA, that portion of the RNA will fall off the DNA. Another molecule we've discussed that has hydrogen bonds giving the molecule important properties are water. You don't have liquid water unless there are hydrogen bonds between the water molecules, unless the water is frozen, in which case you do have hydrogen bonds, but they don't break, and then you have solid water. What would happen to these molecules if the hydrogen bonds were to all disappear? So in one instance, all the hydrogen bonds on the planet Earth were to break. What would happen to these molecules? Well, for DNA, what would happen if all the hydrogen bonds were to break?
The DNA would split apart.
Yeah, you'd have single-strand DNA, which, well, it probably wouldn't go through DNA replication correctly, so it probably wouldn't work. If you had all the hydrogen bonds break in water, you'd have no liquid water. You'd have no gas, you'd only have gaseous water if all the hydrogen bonds were to break. You wouldn't have solid water either if all the hydrogen bonds were to break. All right, so water, amino acids, proteins, and nucleotides and nucleic acids all have hydrogen bonds, and we've talked about all of these in this lesson. Any question about any of that? If not, I'm going to get started on the next lesson just a little bit because I want to work ahead because I don't think you guys are going to want me to cover a lesson, chapter 7, right before you have the final. So let's go ahead and work a little bit ahead. And the next chapter is, I think, chapter 3. We'll only get started, and I promise we will end the lesson a little bit early. Any question about chapter 2 before I close it down? All right, chapter 3, I'm showing you a major goals and a rough outline. Remember, for all of the goals, you should take a look at the objectives for chapter 3. Shoot, I don't have any water there. All right. So know the terms. What's resolution, refraction? What is a stain? What is a basic and acidic stain or a basic and acidic dye? What is a smear preparation? What is a mordant? And what is peptidoglycan? 2, understand the sizes of organisms and the properties of microscopes. 2A, understand the major differences between the light microscope and the electron microscope. 3, understand why staining improves resolution. 3A, know the 3 types of stains. We have 3 types: a simple stain, a differential stain, and special stains. And then 3B, understand the gram stain, its procedure, and the basis of its differential staining. Let me make this larger so you can see it better. The units of measurement in science as well as in medicine, we use the metric system. You are expected to know the metric system for this chapter, so the quiz of this chapter, and you should know most of the things. See if I can get that. That'll do. You should know most of the things on the left side here. You should know a kilometer is 1,000 meters, and that a meter is 100 centimeters, meaning 100 centimeters equals 1 meter. You don't need to know a decimeter. You should know a millimeter. There's 1,000 millimeters in 1 meter. So a meter is 1/1000th of a millimeter. A microliter is, there's 1,000,000 micrometers in a meter. So a meter, let's see, a micrometer is 1/1000th of a meter. And you should also know that the size that we measure under the microscope is around 1 micrometer. And then a nanometer, you should know this one because the size that we measure in an electron microscope is a nanometer. And it is 1/1000th of a meter, or there are a billion nanometers that make 1 meter. You do not need to know a picometer. Any question about any of that? The metric system is easy once you learn it because everything is based on a multiple of 10. Let me see, there's a hectometer and a decameter. So there's 1 meter, you don't need to know a decameter. There's 10 meters in a decameter, 10 decameters in a hectometer. You don't need to know those 2 terms. And then 10 hectometers in a kilometer. So everything is based on a measurement of 10. And we can say there's 10 decimeters, you don't need to know this term, in a meter. And there's 10 centimeters in a decimeter. And there's 10 millimeters in a centimeter. So like I said, everything is based on a power of 10. You only need to know the kilometer, the meter, the centimeter, the millimeter, the micrometer, and the nanometer. The others you can learn if you wish. You don't need to know it. You should also know that in metric, when we're talking about length, we're talking about something based on the meter. So that's the base unit. And then for weight or mass, we use the term gram. And so our base unit is 1 gram. And then you use a kilo for a kilogram. That's 1,000 grams for 1 kilogram. And then we use the centigram for 1/100th of a gram. So we use these prefixes for all the different units. And a meter is a unit of length. A gram is a unit of mass, or is it volume? No, mass or weight. And trying to think what the third one is. Volume. We have a liter, which is the base unit for a volume. And you can say a kiloliter, or a centiliter for talking about the volume. Any question about any of that? So you do not need to know the metric equivalent, and that is, I should say, the US equivalent, and that is there's about 0.6 miles to 1 kilometer. You don't need to know that. And the US equivalent is very difficult. But you do need to use metric and only using metric. But you should be converting from one unit to another. Like if I say something like there's 1,000 centimeters, how many meters is that? You should be able to get me the answer there. And what I usually do when I'm going from one unit to another unit, like a kilometer to how many centimeters, I convert first to a meter because that's the one I have memorized, and then go from meter to a centimeter. Any question about any of that? All right. The second reason why you should know the metric system is this map is showing you all the countries in the world that do not use the metric system. And as you can see, the majority of the countries in the world, 3 specifically, do not use the metric system. And only one of them is a big country, the United States. All the other countries in the world, except for these other 2, use the metric system. And 2 of these 3 are slowly converting to using the metric system. The United States is one of the countries which is slowly converting to using the metric system. And if you look on a modern car dashboard, it will tell you your speed in miles per hour, as well as the speed in kilometers per hour. And oftentimes on the road sign, you can see in big print the speed limit going, 60 miles per hour. And then it'll tell you in smaller print the kilometers per hour you should be driving at. That's because the United States is slowly converting to using the metric system, and we've been very slowly converting to using the metric system ever since Thomas Jefferson passed legislation for the United States to slowly convert to using the metric system. And as you can tell, we've been very, very slow about converting. But you should know the metric system for 2 reasons. First, most of the countries in the world use it. And second, science and medicine use the metric system. Any question about any of that? All right. I think I'll stop here and we'll ask if there's any questions. I promised to stop the class early, so we'll stop here.
Can I ask a question to the class?
Yes?
Would anyone be willing to be my study partner?
You should ask that question in the group discussion. Of course, I've got to open it up, but you should ask that in the discussion and then people can email you.
Okay.
Let me see if I can show you that. Did I just shut down my sharing my screen?
Yeah.
Let me share your screen. Let's close that down. Come on.
Hey, we have a group already set up, and I'm happy to have you join if you'd like.
Oh, okay. Do you
yeah,
need my
if
phone
there's a group,
number?
if the group has the size of 6 or more, I'm going to ask you to form 2 groups of 3. But if it's less than 6, you can add somebody to it.
Me and Lindsay have a group if you want to join ours.
All right, you guys,
Oh,
let's have this discussion in the group area. I'm trying to show you that now. And it's true, I got to open it up. But can you see the screen?
yeah.
Yeah, student discussions. And let me go ahead and open that now because I think it's shut because I've got to grade it. Why didn't that open? Oh, it did. Just Go ahead and make that open to December. Oh, I see what the problem is. Let's just go that week. All right, you can have that in the student discussion. And like I said, if you have a group of more than 6, please divide into a group, group 6 or higher. Please divide into 2 or more groups.
Okay, thank you.
All right.
By the way, I sent you an email earlier, and I was wondering when you might get back to me because it's been a few days now.
What was your email?
What was my email about?
Yeah.
It was regarding the point that was removed from my the Plagy quiz, and I retook the environmental quiz to get it back, but
Oh,
you never
I
responded to
haven't
my email
graded.
about that.
I haven't graded the retake of the environmental quiz yet. Or if I did, I haven't changed the grade in your environmental quiz.
That's like-
So that's aware of to me. It's something I got to do. I don't know when I'm going to get it done.
Okay, sounds good. I just wanted to know if you were aware of it or not. So thank
Yeah.
you.
All right.
Yep.
All right. Any other questions?
I have a question. You have a quiz discussion for this week. Is that like for all the quizzes? Like, do you want us to discuss of when you want all of our quizzes, or is that just for the final?
I think that's already closed, so you can't get credit if you add that now. But I was asking if you wanted the labs to be graded 1 time or 2 times. Is that what you're asking?
No, it was a quiz discussion, and it's for
Quiz
this week.
discussion. I'm not sure. That's probably something that was from previous. Let me take a look at that.
Okay.
All right. I don't believe there is a quiz discussion that I made for this term. So let me look into that. What is it on the grade book or something? Because I'm not finding it here. Oh, there it is.
Okay, I was gonna say
I
it's on the
think that's something
module.
from an earlier term. And
Okay.
so it is not going to be something to do this time.
Okay. Thank you.
Yeah. So let me get rid of that. Thank you for pointing that out. Like I said, that was a for an earlier term. I guess I can get rid of that too. Well, we'll leave it in case I have a quiz this week. All right, any other questions? If not, I'm going to log off. I'll see you at 6:30 for the lab.